One of the really wonderful things about grasslands is that they've been around for a really long time and they hold a lot of history in them. We often think about trees as being the plant species that live forever, and while they do live a long time, one of the little known facts about grasses is that some of them can live up to 100 years old. And grasslands have, when I say been around a long time, like they've been around for, over 10,000 years. And grasslands used to stretch like all through the center of North America. And if you can imagine this like vast expanse of these grasslands, they developed into these very complex ecosystems and provided a lot of intricate ecosystem services. So you have the plant communities, you have the insect communities, you have these grazing communities all working in conjunction with one another. And over time, one of the most dynamic things about grasslands are their soils. So if you can imagine in this tall grass prairie, we see these plants above ground, and what we don't see is their roots that go way deep down into the soil. And in doing so, these grasslands are acting as water infiltration systems and water purification systems. They're acting to harbor nutrients and carbon, which is something that we're really interested in today with climate change. And so over time, these grassland soils really developed into this amazing resource. And humans figured out that they could tap into those resources. And what I mean by that is that it is the soils here in these grasslands that went on to produce what has become agriculture in the Midwest. Essentially, what humans have done for a really long time when they disturb the prairie and start to farm on them is they're essentially mining all of that nutrient work that our grasslands have been doing for that 10,000 years. And so one of the things that I'm really interested in doing with restoration is thinking about how do we not just restore the plant communities, which has been like this historic view on restoration, right? Like we can really take this above ground approach to our restoration objectives. And what I'm interested in is thinking about how do we also restore the below ground community, that is all of these different soil organisms, and how do these soil organisms help to rebuild some of these grassland soil dynamics. And in turn too, that there's like that partnership between what is happening in the soil below ground and what is happening above ground with our plant communities. And this is particularly important because there isn't a whole lot of tall grass prairie left. And in those places that we are able to restore, that soil has been really altered by those past agricultural practices. In addition, these grasslands are facing to this day a lot of challenges. So while it looks like we're standing in this vast expanse prairie out here, what's really happening is right over there is a road. And as you can see, there's actually some road construction going on right now. And so at the same time that we've become more conscientious about the services that tall grass prairie systems provide, we also still face this challenge of human expansion and human land use. And so in those areas where we can go back in and restore the prairie, we're not just restoring just the grassland itself, but also those ecosystem services, and also, I would argue, our relationship to those places. So we've been talking a lot about grassland soils and the potential for these grassland soils to act as these carbon sinks and these nutrient-rich soils. And so I thought it would be a good idea for us to actually get a look at one of these soils. And the way we're going to do that is we're going to take this soil core, which is a pretty simple tool. And what we're going to do is we'll find a space that we want to take a core from, and we just want to kind of push back the plant material a little bit. And then I'll put my soil core down, and this is the fun part. And I just love doing this because it's like, you know, taking a peek at like what's underneath my feet. So I'm going to give this a little bit of a twist. So when I pull up the core, I just took a little bit of a shallow core, but we can see here some of our prairie soil. And what I want us to notice is in part like how rich and dark this soil is. And that's because there's a lot of organic matter in this soil. And that organic matter is really where all of that nutrients and that carbon is stored. So I'm just going to kind of take this core into its pieces here, and one of the things I want to just talk about is the structure of this soil. And so one of the other important aspects of our prairie soil is that it has this soil structure to that. And what I mean by that is that it has this complex system of soil particles that are all fitting together that allow it to have the stability that this soil has, and to allow it to protect those nutrients and that carbon, and to allow that water to remain in that soil longer. So one of the things that I also want us to take a look at right now with this soil is that, you know, in my hands I'm holding soil organisms, and in particular I'm holding bacteria, and I'm holding onto fungi that's in the soil. And so in the same way that like we have a microbiome that helps with our health, the prairie has its microbiome that helps with its health. And it is this prairie microbiome that we're really interested in looking at with regards to restoration. And you may be asking, like, well, what is the, you know, why would we need to think about a prairie microbiome for restoration sites? And the reason for that is, if you think about it, all of our restoration sites are former agricultural lands. And these agricultural practices, like adding fertilizer, of tilling the soil, which takes these aggregates and just breaks them all up and makes these aggregates unstable, because it's just breaking them up over and over and over again and making them smaller and smaller and smaller. So we have, you know, tilling the soil, we have adding these fertilizers, we have the fact that we plant crops in monoculture, and so we're just adding the same plant inputs into the soil year after year after year. And with all of that too comes erosion of the soil. And so essentially what agricultural practices do is they shift the microbiome so that, you know, it is reflective of those agricultural practices, unlike our prairie soils. And so if we're trying to restore that land in the same way that we may try to restore our own microbiomes, we have to think about how do we shift that microbiome back so that that soil and the plant community that comes along with it can function together in unison. And so what my research is really looking at is, like, what are the best ways of really thinking about how to get this prairie microbiome into these places in need of restoration. So now we are standing in what is a intentional restoration. And by that I mean that this land that we're standing here on now was former agricultural land that the Nature Conservancy then decided to restore with native prairie species. And one of the challenges that we have with our current restoration practices is that while we apply these seeds to these former agricultural lands, we actually end up with very low diversity as compared to our prairie systems, which, as you saw earlier, had just a ton of different plant species. And as we look across this particular field here, we see, upwards of like 5 to 10 species only. And I'd also like to point out that another challenge just with restoration is that you have this invasive plant encroachment. And so here we have what is called Lespedeza cuneata, and this is a species that will often move into restoration sites and end up taking over whole entire patches of the restoration. And so the question becomes like, well, why are these restorations vulnerable? And also, why is there such low diversity in these prairie in these prairie restorations? Why can't we get them to this diversity that we have in our remnant prairies? And the other thing to consider with that, right, is like we're talking about that above ground system, like measuring that from this above ground approach by thinking about plant diversity. But what plant diversity also reflects is the diversity below ground and the ecosystem processes that are happening below ground. And so that is a concern, like the land above ground, if we go back to thinking about story, that land above ground is telling us the story of what's happening below ground. So we have these challenges. And what my research is really looking at, again, is going back to that microbiome, is like asking the question, well, what is missing from the microbiome in this restored site? Is there something, is that microbiome asleep? Does it need help waking up? Are there members of that microbial community missing, as we talked about with those agricultural practices? And so in that, we're really trying to think about how do we reintroduce a microbiome to a system, and what are the effects of introducing that microbiome to the system on both plant diversity and our ecosystem function. So if we go over here, this part of our restoration looks very different. And in fact, it looks like our prairie. And surprisingly, it is our prairie. And the question is, well, how did it get here? And this is really what in part, what my project is about. So as we saw earlier, there's some road construction going on. And what we were able to do was leverage that opportunity to actually move giant chunks of prairie that we refer to as monoliths and insert them into different areas of this preserve that are in need of restoration. And the way we did that was by using a giant tree spade to move these monoliths that are 1.5 meters wide by 1.5 meters deep. And that allowed us to move both the plant community and the microbial community and all of those great soil ecosystem functions that the prairie provide. And so essentially what we're doing is we are introducing a microbiome to this restoration site that is in need of help with its microbiome. And in addition to that, what we did was we thought about, well, what is the best concentration in which to introduce a prairie microbiome? And we did that by placing different concentrations of these prairie monoliths together. And so we have monoliths that are 6 individual monoliths that are spread apart. We have monolith clusters that are medium in size, and then we have a monolith cluster that is really large in size. And so the idea there is to think about how does the concentration of an introduced microbial community influence both the resilience of that microbial community and how that microbial community is able to spread into and enhance the restoration site that is around it. So that recipient site, it's receiving that microbiome. And so with that, we're studying a number of things, from looking at who is there in the microbial community, both inside and outside of the monolith. And we're also taking a look at how the response to the plant community and also the soil properties are changing with regards to being in the presence of the monolith itself. So one of the things agricultural practices and their microbiomes. And so what my work is also thinking about is like, how do we put this system back together? And this is really important because the plant and the microbiome together are essentially the foundation for our grassland ecosystems. And it's a really amazing relationship because essentially it's linking this below ground and above ground component together. And in that way you have this total ecosystem working together. And there's several ways of thinking about how to get at if these different size concentrations of monoliths are doing the job that we want them to do. The question being there, like, is that prairie microbiome able to spread into the restoration site? And so here we're looking at one of our large patches of prairie monoliths. So we have 6 monoliths that are all joined together to create this big patch of both prairie plants and their microbiomes. And with that, one of the ways that we're looking at the spread of the microbiome is to use the plants themselves as potential indicators of if the microbiome is spreading. And so I want to focus in on these 2 plants. So here we have a species of liatris, and we also have this species here, which is Rudbeckia. And these plants that I'm pointing to here are planted just outside of the monoliths, but to note they are planted inside of the monoliths, and they're actually planted really far away from the monoliths too. And so what we're doing is we're actually using the plants themselves to tell us that story if the microbiome is moving out. And the way we do that is we look at different measurements of these plants. And an important thing to note is that these 2 plants here have very different relationships with their microbiomes. We often see Rudbeckia at what we would call like an earlier stage of a prairie. So Rudbeckia might come up in a prairie after it's been heavily disturbed either by fire, or say a bison comes wandering through and rolls in the grasses and disturbs the soil. You may see Rudbeckia appearing there first. And one of the things that we know about Rudbeckia is that it's not as responsive, say, to its microbiome. So it can do okay without it if it needs to. And that is really contrasted with liatris, this plant that we see here, where liatris really forms a partnership with what we call arbuscular mycorrhizal fungi. And these are fungi that form a mutualism with liatris to provide liatris better access to water, better access to nutrients, maybe pathogen resistance, and in turn liatris is providing carbon to that fungi. And so this plant is really dependent on its microbiome, and in particular these arbuscular mycorrhizal fungi, for their growth and their ability to establish, grow, and also produce seed for its future. So we can use these 2 plants juxtaposed to each other to get a good idea if the microbiome is moving out of the prairie. And that would be, for example, indicated by how well these plants are growing against like how they're growing, say, inside the monolith versus really far away from the monolith. So we might think about a hypothesis for that, and the hypothesis would be that the liatris would grow better actually inside of the prairie monolith than, say, far away from the prairie monolith. And at the edge, hopefully it's starting to look more like what it would look like if it was growing inside of the prairie, because that would be an indicator of that microbiome spreading outside of the prairie monoliths. So I want to welcome you guys to this old field site, and I call this an old field. You'll hear ranchers and farmers also refer to it as a go-back field, and what that essentially means is that this was a former agricultural field. This one in particular was last farmed about 16 years ago. And unlike our restoration site, there hasn't been any like intentional restoration done to this site. And So as you can see, if we look across this field, like all these plants are here because they just sort of like found their way into this site, or they're from the seed bank. And they represent more of what we would refer to as like weedy species, which are species that maybe dominate a site after disturbance without that intentional restoration. And we also have some other problems that we will have with our old field sites. So for instance, we also have that Lespedeza cuneata that we talked about earlier in the restored site. And we also have a lot of what we call woody encroachment in this site. And so that is a real challenge for both prairies, but especially these old fields that aren't managed for, for restoration or for bringing them back to this like intentional prairie system. And what will happen if there's not that disturbance of fire, or there's not a disturbance of like our bison grazers, or even the disturbance of the lack of the microbiome that we've been talking about, these woody shrubs start to move in, and that can really severely like alter the system. And the longer we let these fields go, the more they struggle to actually go back to or be reflective of prairies and the prairie system that we've been talking about today. Here we are now somewhat familiar with these prairie monoliths that we were talking about earlier, and these prairie monoliths are located in our old field site. And I just want to note a couple of things. Like, you can really see the contrast here between our prairie species and our prairie system and this old field and our old field system, and the ways in which these two systems may be interacting with each other. And this one in particular is a cluster of 3, so it's not as large as that 6 group that we had looked at earlier, but you can still see the diversity of the prairie represented in this medium-sized cluster. Earlier we looked at the soil from the prairies, and I just wanted to give you guys an idea of what our soils in an old field may look like. And remember, like, this old field holds on to that soil legacy in the soil itself. And we will actually be able to see that here in a moment. So I'm going to use our soil core again, and I'm going to take a core. There's more compaction. See if we got enough here. I can always take another one. So some important things to note about this soil core. Like I said, there's like more compaction, but as you can see, like, this soil core is a lot lighter than the soil core I showed you earlier. And there's several reasons for that. The first one is that this soil does not have that rich organic layer, right? That organic matter layer that our prairie soil have. That's all essentially been, harvested off through agriculture, or has, run off because of the soil disturbance. And so we have left is like this soil that was below that organic matter, but also a soil that has been impacted by that soil legacy. And, these sites are not that far apart, so they've experienced the same weather conditions as, from here to where the prairie was. And yet you can see how dry this soil is. So it's not hanging on to that soil moisture, so it's not doing a good job of retaining that water. And as you can see, this soil is just a lot more crumbly, so it doesn't have that soil structure that we were talking about, which is in part responsible for retaining that soil moisture and that organic matter and that nutrients. And you can see as we go down through, these different parts of the soil core, that this soil just has a lot more clay in it, and it's just not as stable. And with that instability, like, it's basically like that cycle, turns on itself, and we get again, that less soil moisture, less nutrient retention. And again, really importantly, we have a different microbial community in that soil core than we have here. And one thing we haven't talked about yet is that, we talked about the relationship between fungi and the plants, but another thing that fungi do is they really do help with that soil structure. And soil structure is really the base for that nutrient and carbon retention in the soil. Okay, so we've talked a lot about these agricultural practices like adding nutrients, for example. But one thing that's really important to think about is how those land use practices like tilling, for example, how when we till the soil, we are altering those microbial communities by actually like cutting through these different networks that microbial communities will create, and that'll in part affect the soil. And in addition, just the act of tilling itself, when we talk about that soil structure, that the act of tilling and driving over those fields, it results in this soil compaction, which basically has like this, it may just be soil compaction, but soil compaction has so many other effects to the soil community itself, because with soil compaction you have, soil runoff, soil erosion, and again, that's really going to affect like what kind of microbial community is going to be there resulting from these agricultural effects. So compaction is a really big issue, especially when we start thinking about how to restore a field. Like, how do you uncompact soil? And what we know about, say, soil fungi and like their, what we call their hyphal networks, is like they really benefit soil structure, and so that may be a way to essentially like uncompact soil in the long term. say to define soil compaction,
Yeah,
and then you can,
great.
okay.
Whenever you're
Okay. So to define soil compaction, if you can imagine these big machines, right, that are driving over these fields, and they're essentially all the weight of that machine is literally pressing down on that soil. And because we've like gotten rid of that organic matter and there's no cushion left. And so the more that we till and the more that we drive over these fields, the more compacted, the more compressed these soils get. And it should also be noted that like through that tilling process, you know, we're going down in the soil as deep as that equipment is allowing us to. And so it can have like this compaction effect, can then like ripple down through the soil itself. So even though, you know, we're just thinking about this machine driving around on top and pressing all of its weight, that pressure, that compaction, it goes down through the soil, you know, several feet. And that again can make restoration a real challenge.
Whenever you're ready.
Okay. We've been talking a lot today about microbial diversity, but what exactly does that mean? And, you know, what does this prairie microbial community potentially have that this old field community does not, in terms of how this old field community might be able to support prairie restoration? So here we have representations of what microbial diversity might look like. So as you can see here with the prairie, we have a multitude of species, and that's represented by both the different colored colors and also the different shapes of this microbial community, but also how many of that certain individual within the microbial community exists within the community itself. So that's like the notion of diversity. Versus in this system here, this is an extremely low diverse microbial community. We only have one species and that's it. So when we think about this like prairie restoration, the idea is what can the shifts in these microbial communities bring to the soil and to their partnerships with the plants that we talked about earlier. So for example, if we have this arbuscular mycorrhizal fungi represented, say, by this blue species or this black species, and it has its mutualist host of a prairie plant, it may be able to forage its way out into this old field and provide nutrients for its plants in the prairie, and at the same time provide diversity and also ecosystem functions to this old field that were not present before. And so over time we may see a shift in this microbial community in this old field. That's what our hope is. And so we'll see some of these community members be able to enter into this community, which, as you can see, there's a lot of room for growth here with this microbial community.